where did we end? I think we end at amino acids, something like that. Proteins, yes. And where did we start?
Biomacromolecules.
Okay, Biomacromolecules. Okay, this is where we start. Okay, so a quick summary about you, Hiram. Are you okay?
Yeah.
Yeah. Okay. Let's go with a quick summary. Tell me,
Yeah.
what did we do in the last class?
Biomacromolecules. A common feature of all compounds in acid-soluble pool is that they are small. Their molecular weight ranges from 18 daltons to 800 daltons. Bigger molecules, which are macromolecules, comes in acid insoluble pool. Those biomolecules which has molecular weight less than 1000 dalton, those are called as biomicromolecules. Those biomolecules which are found in acid insoluble fraction and whose molecular weight is above 1000 daltons, known as biomacromolecules.
Yeah.
The acid insoluble fraction has 4 types of organic compounds: protein,
Yes. So when we use the word acid insoluble fraction, or we use the word biomacromolecules, it's same, right? It's synonymous. I think it's clear, right? Okay, continue.
So they are proteins, polysaccharide, nucleic acid, and lipids. Molecular weight is in the range of 1000 dalton and above. They are polymeric substances.
10,000 daltons.
Yeah, sorry, 10,000 daltons and above.
Yes. We're just making it very small. 10,000. Don't underestimate the power of a protein. Yes. And what's the problem with lipids?
Yeah, so a lipid whose molecular weight is less than 800 Dalton still comes under acid insoluble fraction, macromolecule fraction. This is because lipids are smaller in terms of molecular weight, but they are arranged into bigger structure inside a cell, like cell membrane and other membranes. Even when we grind the cell, break down the cell, these membranes are broken down, but in the form of vesicles, which are not water soluble, and hence these membrane fragments still belong to acid insoluble pool.
Yeah, so if I ask you, so does it mean that lipids are macromolecules?
Well, partially we can say, they're not strictly.
No. Lipids are not macromolecules. They just behave like macromolecules because they always live together. Okay? They always are found together. You understand?
Yes.
So just remember that lipid is not a macromolecule. We studied lipids in biomicromolecules actually when we studied amino acids. But here we are studying proteins. So amino acids become proteins, sugar became polysaccharide, nucleosides became nucleic acids. Lipid are still lipids. Okay, because they behave like biomacromolecules. Okay, but they are not macromolecules. Yes, continue.
Yes. So the chemical composition of a living cell, water is 70 to 90%. Proteins are 10 to 15%. Nucleic acid is 5 to 7%. Carbohydrate is 3%. Lipid is 2%, and ions are 1%. Then we started with polysaccharide. These are simply long chains of sugar molecules. They are like thread containing different monosaccharide as a building block. Example, cellulose. It is a polysaccharide made up of only one type of monosaccharide called as glucose.
Yeah.
A LOMO polymer and a polymer of
Homo, not LOMO.
homo.
What's LOMO? Iram
Okay.
is already devising new. You are
So I
just
just wrote
increasing the
LOMO.
you are increasing the syllabus, Iram. I am trying to keep it simple. What we know as scientists. Iram is finding out LOMO polymers, polo polymers. Okay, anyways, good. What do you mean by homo? The word homo means similar, right? Homogeneous mixture, which means a mixture which is similar. I think we've discussed this, right? So homopolymer is a polymer which is made up of same kind of monomers. So cellulose, also starch and glycogen are homopolymers. Okay. Even inulin is a homopolymer made up of fructose. Okay. Yeah, continue.
A polymer of fructose is also known as inulin. Then we have plant cell wall that are made up of cellulose, chitin, which is exoskeletons of insects, arthropods, and complex polysaccharide are also there. There are polymers of nucleotide, which is phosphoric acid, monosaccharide, and nitrogenous bases. Nucleic acid together with protein and polysaccharide forms the true macromolecule of a living tissue or a cell. In a nucleic acid, one purine base makes pairing with another pyrimidine base. As a sugar
Yes.
from nucleic acid is either ribose sugar or deoxyribose sugar. Ribose sugar is found in RNA, and deoxyribose sugar is found in DNA. Then we have proteins. They are polypeptide, long chains of polypeptides. The long chains of amino acid which are linked together by peptide bonds. Protein chain
Yes.
is always a heteropolymer, not a homopolymer,
Why?
because they are different. I mean,
Okay, smart answer. Like, what is a processor? A processor is a processor that processes a process. Because there are 20 different kinds of amino acids, right? And in our cell, and every protein contains more than one type of amino acids. So the monomers become different. So the chain become a heteropolymer. Okay.
the amino acids which are not important or essential for our health and which cannot be synthesized by our body, making it very important to supply through adequate amount of essential amino acid. There are 9 essential amino acid: um, histidine,
Hill TV MTP.
HLTV-MTP,
Yes, histidine,
isoleucine, leucine,
isoleucine, leucine.
isoleucine.
Hmm.
One minute,
There's
sir.
there's one there's one more L. I made you write this.
Yeah,
Yeah.
isoleucine, T for tri.
Hey, there is one more L. Histidine, isoleucine, leucine. Which is the other L? There are two L's I can see. Aisha, would like to help you?
Lysine.
Lysine. What?
Oh yeah,
Isoleucine, leucine, and lysine. Yes, good. And then TV is
tryptophan, V,
tryptophan. V
valine,
is
valine.
valine.
M
Yes. M
is
is methionine.
methionine. T
Yes.
is threonine, and P is
Threonine.
phenylalanine.
Phenylalanine. Yes, perfect. Okay, then. So essential amino acids, we have to get it from the diet
Right. Then
always.
there are non-essential amino acid. These are synthesized in body through some or other metabolic pathways.
Yes, so no need to, even if you do not get it through diet, if you get other amino acids, you can convert other amino acids into this one. So they do not become essential, they are non-essential. So you just need to take care of the essential amino acids first. The non-essential ones will be synthesized in the body, right? Okay, very well. So today we will be talking about this major topic where we'll go to structure of protein. So we are studying proteins, right? And it's not yet over. So we will start with structure of proteins. I told you that a protein is not functional until it is properly folded into a three-dimensional structure. Yes or no? Yes. Just a long chain of polypeptide of amino acids is not going to make a protein functional. Okay, so write down a protein is functional in the cell protein is functional in the cell only when it is properly folded into its three-dimensional shape. Is it clear? So even if you make a right polypeptide chain, but you misfold it, it's a waste. That protein will again be, the cell will retry, the mechanisms in the cell will try to refold it into proper shape. If they cannot refold it, then they will destroy the protein. So just by making, but it happens, it does not happen very often. Mostly proteins are folded in a very controlled way, coordinated manner by another group of proteins called chaperones. Chaperones are the molds for proteins. But sometimes if they are misfolded, they do not, they become non-functional, or they have some other functions, they misfunction. So for that matter, either you have to fold it correctly or you have to degrade it. You understand, Hiram and Asha?
Yes, sir.
Yeah. Now the structure of protein, write down, the protein structure is described at 4 levels. At 4 levels. First is called primary structure. Primary structure. And what do you think is a primary structure? It's simple. It is simply the sequence of amino acids, right? In a primary structure, a protein is not functional. Okay. So first you make a long chain. So write down, primary structure is just the sequence, the linear sequence, the sequence of amino acids. Okay. And what does this provides you? What information does a linear structure gives? It just gives the structure of the positional information of every amino acid in a protein. It provides the formation of amino in a protein. Okay. You understand? So if this is amino acid, then it will be connected together in a long chain like this, where these are amino acids and these are peptide bonds. Okay. Simple, the primary structure. Clear to both of you, Aisha and Iram?
Yes.
Okay. And the first amino acid, which is this one, it starts from first and goes towards this. The first amino acid is known as N-terminal amino acid. Okay, nitrogen terminal. Can you tell me why? In every polypeptide chain, the first amino acid is known as the N-terminal amino acid. Go back to the structure of amino acid in your notebooks quickly and see that on one side there is NH2, right? And on the other side there is COOH, carboxylic acid group. So there is an amino group on one side and there is a carboxylic acid group on the other side. Am I correct? In the center there is alpha carbon, towards the bottom is a hydrogen, and on the top there is a R group that is variable. Yes or no?
Yes.
Yes. So the N terminal is called because of amino, nitrogen. So on one side is the nitrogen, on the other is the carboxylic acid. So the beginning of every amino acid, where it begins, towards this side is the nitrogen in every amino acid, and towards this side is the carbon in every amino acid. So the last amino acid will be called C terminal amino acid. Okay. Last amino acid in a polypeptide chain. Is it clear? Say yes or no?
Yes, sir.
Okay. Now this primary structure first folds into a secondary structure. Now if you have a long thread, a long polymer, you can either fold it secondary in the form of a helix, right, like a DNA folds, right, or beta pleated sheets. So you can do two things. Write down secondary structure. This is the second level. Secondary structure of proteins. Write down. The long protein thread can further the long protein thread or polypeptide can further be folded into first alpha helix. Okay, helical shape. So this long chain can either be folded like this. You understand? So there will be amino acids. Is it clear?
Yes.
Yeah. So this is the alpha helix. And in protein, only right-handed helix are found. This is an entrance question. This is very curious, but somehow only right-handed helix are found. No left-handed helix is there. Okay.
Sir, right-handed helix means
Right-handed helix means you take the thread and you make the helix in a right-handed fashion. So if you look from the top, it will look like it is coiling in a clockwise manner, not in an anti-clockwise manner. You understand? If not, let me show you through cable of my adapter. Can you see this cable?
Yes, sir.
Yeah. If I start folding it in a right-handed manner, so this is a right-handed fold. I'm just making a helix in a right-handed manner. Can you see the right-handed fold? I'm just rotating through in a right-handed fashion.
Yes.
Okay. If I go opposite, if I do it in a left-handed fashion, then it will look something like, it's not looking like it, but it will look something different. Do you understand? So either a right coiled manner or a left coiled manner. So if it is right, from the top, when you will look, it will look like a clockwise pattern. But if it is left, it will look like anti-clockwise. Okay, clear? Aisha?
Yeah.
And Iram? Yeah. So in proteins, only right-handed coiled helix are found in the secondary structures, no left-handed helix. This is called alpha helix. Or second thing that can happen with proteins is beta pleated sheets. Beta pleated sheets. Now this looked something like this. So you have a thread. Instead of making a helix, you put it into sheets. You understand? One after the other. Again, I'll use my cable. I hope it works after all this experiment. So instead of making a helix, we are compacting it together by making sheets like this. Can you see this sheet?
Yes.
And actually, I have seen people compacting their charger cables like both. So you see this? This is not a helix now. Is it visible to you?
Yes.
This is like a sheet arranged in sheet. It is thin and it is arranged in sheets, no helix. Yes. So a thread can be folded as a alpha helix or a beta pleated sheets. This is the structure. Okay. Is it clear? In a protein structure, it is never the case that either only alpha helix is there or only beta pleated sheets are there. Both are present in some or the other proportion, because protein is a long chain, no? So what will happen is, again, I'll use the cable. I don't think it's going to work after tonight, but yes. So it's a long cable. So first half will be like in the form of a helix. Can you see this is in the form of a helix like this?
Yes.
Yeah. And the later half then can be pleated in sheets. Okay, like this. So both things can be present in the same structure like this. Can you see? One is helix and the other is sheet. Yes.
Yes.
You understood? Very good. So both beta secondary structure, which is pleated sheets, and alpha structure, which is helix, are present in the secondary structure. After that, we have the tertiary structure, the third level. What do you think happens in a tertiary structure? Both alpha helix and beta pleated are present together, and they roll into a spherical ball to make protein look more spherical in structure. Is it clear?
Yes.
So write down, in addition to secondary folds, if the protein chain if the protein chain also folds upon itself, to look like a hollow ball or a hollow woolen ball. Woolen has just one single L. You must have seen balls of wool, right?
Yes.
Woolen balls, which grandmothers used to knit the sweaters, right? Or mothers also. It's not like only grandmothers can knit. But woolen balls are not hollow from inside. Inside also there is wool. Now imagine a woolen ball which is hollow from inside. There's nothing inside. It's just like folded upon itself to give a spherical structure. So that will be the tertiary protein. Hollow woolen ball. Then this is the tertiary structure. That's a little different for me to demonstrate using the wire, but you understood, right? Yes, Iram, thank you so much for saving my data cable. So this is tertiary. Now these three, primary, secondary, and tertiary structures are there in every protein. No matter what the protein is, these three structures will be for sure present. The protein will go through first the primary structure, which is just the linear chain. Then it will fold either into alpha helix or beta plated sheets. That will be the secondary structure. Then it will fold upon itself to give spherical shapes or other kind of shapes, tertiary structure. Sometimes proteins also have this one additional structure known as quaternary structure. So the fourth is quaternary. Quarter means 4, right? 1/4. So quaternary structure. Write down. Some proteins are an assembly of more than one polypeptide chain. More than one polypeptide chain. Okay. And some proteins are an assembly of more than one polypeptide chain, or in the bracket write down more than one protein subunit. More than one protein subunit. Okay. Then the protein structure is known to be a quaternary structure. Protein structure is known to be a quaternary structure. A very famous example is hemoglobin. Human hemoglobin protein. Okay. So it is made up of 4 subunits. In which there are 2 alpha subunits and 2 beta subunits. Total how many units? 4 subunits. So every subunit will go till tertiary structure, and then all these tertiary structures will come together to make a quaternary structure. Is it clear? But this does not happen for all the proteins. This happens in some proteins only. Is it clear? Hiram, Aisha, please note it down and then let me know when you're done. Okay, so Aisha and Hiram, there is slight change in your schedule from the classes like that will happen today onwards. So you join the classes at what 6:30 KSA, right?
Yes.
Yes, 6:30 KSA to 8:30 KSA. So the revised time, we are shortening your classes a little bit from 6:30 KSA to 8 KSA, not 8:30. Okay, is it clear?
Yes. Sir.
Yeah. So like as we'll move further, you will also get test series that we have to do. Aisha is so happy. Oh my God. I don't know whether I should be feeling happy or sad about it, but Aisha is like, I was already bored to death in the classes.
No, sir.
Right?
Actually, I was about to tell you about like reducing the class time.
You,
there.
you were about to tell.
Yeah.
Okay. Okay, that's called sheer luck. Anyways, but yes, so we're thinking of, so the motive behind shortening these classes is that also to give you some time to focus on your whatever I'm teaching. You should have time to revise it, so that half an hour you can utilize to revise, which I know that many a times you will not. You will take it as a freedom time, but believe me, it will help you when the test series and other tests will start, and even your own examinations will start. That time will help you. If required some time, we can use that time again, but yes, so it will be 6:30 to 8, right? Okay.
So,
Yeah,
so we have exams coming up in September, first week.
September first week. And what's your syllabus?
They just said to prepare 4 chapters, which is cell,
Which is
cell division, morphology,
done.
and
Morphology and
anatomy.
anatomy. Okay, so I can do one thing. Let me finish protein today. Okay, and instead of going to metabolism, so I'm leaving the enzyme and metabolism part of this chapter. We'll come back to it later, and from the next class what we can do is we can start with the morphology of flowering plants. How about that? Because if this chapter is not coming to your exam, all that you needed to know about proteins and all the biomolecules, the basic, you know. Now the higher concepts, which are enzyme reaction, or enzyme kinetics, and metabolism, that we can take after morphology and anatomy. Will it work for you? Because enzyme kinetics is a big topic, and if it's not coming, if it will come later, then we'll deal with it later. Okay, Aisha, I think we have to be flexible with along with your syllabus. So from next class onwards, I'll start with morphology. Cool. Okay, very well. So now today let's focus on protein structures and try to finish it. Now, one thing that will also help you in anatomy. So clear, quaternary structure is clear to you? Now, let's now come to the bonds. So we have discussed different type of polymers made up of monomers, but whenever there is a polymer made, the monomers are linked together, right? So in biology, there are what are the different kind of bonds? Okay, these are chemical bonds, kinds of bonds that link monomers into polymers. So tell me, what all polymers have we studied till now? What all polymers we have studied till now? First we have studied is the protein, right? Protein is a polymer. Polypeptide chain is a polymer, and I've already told you what is the bond that is present between two amino acids. Just know it. Peptide bond, right? But what is a peptide bond? Where it is formed? So write down. A peptide bond is formed when the carboxyl group, okay, when the carboxyl group of one amino acid, so carboxyl group is C double bond O OH. This is carboxyl group of one amino acid reacts with the amino group, which is this group, right? So this is amino acid 1 and this is amino acid 2 in a chain, and now there has to be a bond formation. So what will happen? Do you see this H2O? So let me make it like this then. So there will be one hydrogen here and one hydrogen here, right? NH2. You understand, right? And what is this? If I remove this, what will be removed? H2O released. So water is released. H2O, right? And this carbon forms the bond with this nitrogen directly. And this is known as the peptide bond. So write down. When the carboxyl group of one amino acid reacts with the amino group of the next amino acid, leading to elimination of water molecule, leading
Sir,
to
can you repeat it?
Yeah, I will try. What did I say? When
the carboxyl is
yes, when the carboxylic group of one amino acid reacts with the amino group of the next amino acid, leading to release or elimination of a water molecule. Okay, so this process where you release the water molecule is known as dehydration, right? When our body undergoes dehydration, what do we lose? We lose water, right? We say, no, this person got dehydrated. Yeah. So similarly, when these two molecules together get dehydrated, so they lose one water combined, H2O. And because of the dehydration, this reaction is known as dehydration reaction that leads to the formation of peptide bond. Okay. And this is how it happens. H2O is released. And then carbon joins with nitrogen. Clear? How is a peptide bond formed? It's a dehydration reaction bond. Draw this. Shall I move ahead? Iram, Aisha?
Yes, sir.
Second, what kind of polymer you have studied apart from polypeptide? We have studied polysaccharide, right? Like sucrose, starch. And what is the bond that links the monomers of a polysaccharide? What is that bond called? In this, the monomer is monosaccharide, right? So monosaccharides are linked by glycosidic bond. Glyco word is for sugar. Glycosidic joining bond. You just have to remember which bond is present where. This is peptide bond, this is glycosidic bond, and a glycosidic bond, just write down. In this, you don't have to need the structure, but just write down. This bond, glycosidic bond, is also a dehydration. Is also a dehydration reaction. So here also, what is happening? Elimination of water. Okay, and write down. Glycosidic bond is formed between glycosidic bond is formed between two carbon atoms. Between two carbon atoms of two adjacent monosaccharides. So the difference is it is happening between two carbon atoms. Peptide was happening between one carbon and one nitrogen. Okay. Is it clear?
Yes, sir.
Yes. So that's the difference that you have to remember. Written? What else? What is the other polymer we have studied? We have studied nucleic acid, right? DNA or RNA. What is it? A polymer of nucleotides, right? So write down. Uh, the monomers, nucleotides, are linked by phosphodiester bond. Phosphodiester bond. Okay, phosphodiester bond. Now what happens in this is, how is this bond formed? Write down. How can we, should I make the diagram or write down the phosphate group. You remember the phosphate group, both of you?
Yes.
There was a phosphate group in a nucleotide, a phosphate group, a sugar and nitrogenous base coming inside, right? So the phosphate group links, phosphate group links 3' carbon. So the carbon which is at the third position, 3' carbon of 1 pentose sugar Am I audible, Asha?
Yes, sir.
Where did Iram go? Sorry, I just, there was some network issues. It's fluctuating. Can you inform Iram to join back? Okay, she's back. Hello, Iram. Am I audible and visible?
Yes.
Yeah. So it might happen again. If it happen again, like I'll be joining back in a minute, not more than that. Okay. Sometimes the network fluctuates at my place, so don't go. Okay. Cool. Yes. No. Any emoji will work. Very good. Yes. So we're writing about phosphodiester bond. So write down how it is formed is the phosphate group, which is present in the nucleotide. It links the 3' carbon of 1 pentose sugar to the 5' carbon which means the carbon present at the 5th position of the pentose sugar of another nucleotide. Okay. So basically in one nucleotide, we have 3 things: a phosphate group, a pentose sugar, and a nitrogenous base. The same 3 things will be in another nucleoside: a phosphate group, pentose sugar, and the nitrogenous base. So this phosphate group is shared, common. and it links basically these two pentose sugar. The 3' carbon of one will make a bond with the 5' carbon of the other, like through the phosphate group. You understand? So these two sugars are connected through phosphate group, and because write down, the bond, so this is what happens, and the bond between the phosphate group and sugar is known as ester bond. Now you have to tell me, if the bond between one phosphate and one sugar is called ester bond, then what will be the bond between one phosphate which is attached to two sugars will be called? So if there is one phosphate, it is bonded with one sugar. This is ester. On the other side also it is bonded with one sugar. Then what will be this called? If one is ester, two will be what?
Asters?
Wow. Two will be diester. In biology, we don't do plural by just putting s. That's English. In biology, we use words like mono, hybrid, cross, di, tri, tetra, penta, hexa. In chemistry also, because if you said esters, for 2 also you will say esters, for 3 also you will say esters, for 4 also you will say esters. So one is ester bond. So one is ester, one is ester. So together they will be called diester. You understand where does the name comes from? Phosphodiester. Huh? Yes or no? And because it is mediated by, the bond is mediated by phosphate, so these two bonds are called phosphodiester bond. So basically one phosphodiester bond joins two sugars through a phosphate to each other, and this is what happens in nucleotides. Is it clear? Yes, no?
Yes, sir.
Okay. Any other polymer that we have studied in biomolecules till now? Polysaccharide is done, polypeptide is done, nucleic acid is done. Any other polymer that we have studied? Can you think of any other? No, right? Okay. No, right? Okay. So just write down everything. Let me know when you're done. I will take one last topic from this chapter, which is the secondary structures of nucleic acids. And we will end there. And then from next class, we'll start morphology of flowering plants. Enzyme kinetics, I'll teach you later on. Because this secondary structures of DNA will also help you to understand cell division, where the chromosome condensation happens. So this is a connected topic. Okay, while you're writing, I'll just be back. I'll just get myself water, okay? Aisha Neeram, I'll be back in 1
Yes,
minute.
sir.
Okay. Hey, am I audible and visible?
Yes, sir.
Very good. Done?
That's it.
Okay, so let's go to the last topic of today. That will be, I can sense Aisha is already happy. Last topic, yes. I used to be very happy when there used to be last topic started in a class. But my teachers were very intelligent. They will say a last topic, and the last topic would be the longest, you know, which is the case here also. Okay, so write down. Since we are talking, no, I'll finish at 10:30. Don't worry. How much time do we have? We have 20 minutes. We'll finish in 20 minutes. Because we have studied, so we are in the flow of studying secondary and tertiary structures. Since we have studied already secondary and tertiary structures of protein, also write down secondary structures. You write down characteristics of secondary structures. Characteristics of secondary structures of nucleic acid. Nucleic acids. Here we'll not go to some high ordered other structures. That's not a part of your, that's not in your syllabus, but just know the secondary. So what is a nucleic acid? Clearly a DNA or RNA, whatever it is. What is it? It's a long polymer of nucleotides. Yes or no? Will you call it a homopolymer or a heteropolymer? Aisha will answer.
Homo.
Sorry? Homopolymer. I was just about to say, if Aisha answers correct, then the class will end at 10:30. If Aisha answers incorrect, then it will end at 11. Or I mean 8:30 your time, or at 8 or 8:30. But now I think it's good that I have not said that, because it's not homopolymer, Aisha. It's heteropolymer. Can you tell me why it is heteropolymer? Because I know how you are thinking. You are thinking it has a phosphate and the same sugar repeating, so it must be a homopolymer. But you are missing something that changes, that are of different types. Can you tell, Iram?
Space.
What? What did you say?
Nitrogenous bases.
Yes, correct.
I said
You
that.
said nitrogenous bases, yeah? I heard space. And I was like,
Even
what?
I heard space.
Yeah, I heard Aisha is saying that the space is different between two monomers. Okay, nitrogenous base. Correct. Yes. Nitrogenous bases are of 4 different kinds: adenine, guanine, thymine, cytosine in a DNA. And in a RNA, instead of thymine, there is uracil. So that keeps changing. So a nucleotide is made up of these 3 things. So you just remember that they are heteropolymer. Okay, Aisha, will you remember that? Yeah.
Yes, sir.
Okay. You're not getting the penalty of half an hour extra class today, but think before you answer. Okay. Now, the first secondary structure, write down one of the secondary structures that you have already, we have not studied about it, but we know. So one of the secondary structure is, one of the secondary structures exhibited by or shown by DNA is double helix. You know that, right? We already know this. Yes or no? Say yes or no. So we always make DNA like this, right? Yes.
Yes.
And there are bases which are connected like this, correct? So So this is our DNA is must have made or seen in the books. So this structure is called double helix structure because there are two strands and they both are helical, arranged in a helical manner, spiral manner. So this is the double helical structure of DNA. Okay, and this was given by, it was proposed by two scientists. One was James Watson. You just, if you only remember the name Watson, it's enough. James Watson and Francis Crick. Okay, they proposed this double helix structure. Is it clear? And he also said that these two strands that you see, they have opposite polarity. What do I mean by opposite polarity? Is that, so one strand will be 5' to, let's follow this strand, to 3'. Then the other will be 3' to 5'. You understand? So write down this model, the double helix model of DNA says, the double helix model of DNA says that DNA has two strands of polynucleotides, that DNA has two strands of polynucleotides and both strands runs in opposite direction. And both strands run in opposite directions. In the bracket, write down anti-parallel polarity. Anti-parallel. Alright, why is it not getting erased? Okay. Anti-parallel polarity. You understood what is anti-parallel polarity of strands now? Because this is one thing that can be asked. Okay. And I have already told you that the backbone is made up of sugar and phosphate. Yes, when I was telling you nucleotides, did I tell you this? Yes or no? Hey, say yes or no. If no, then say no. If you have any doubt, if you don't remember, just say no. It's very convenient. No, I'll tell it again. Very good. See, Aisha is the most intelligent one. She's like, say no. Tell us. Let's say I'm unwinding the helix. Okay. Helix is right-handed coiled. I'm unwinding it so that you can see it like this, right? So you know that this thing is the base, right? Which is going towards inside. And here in this backbone is present your phosphate and sugar. You understand? So phosphate and sugar makes the backbone of the chain, polynucleotide chain. Write down, phosphate and sugar makes the backbone of the polynucleotide chain. in a DNA or RNA? Both. Okay, so this backbone that you are seeing, this one, this is made up of a phosphate sugar. Okay, exactly what I was telling you here. See, sugar, phosphate, sugar, then there will be another phosphate making a bond, then another sugar. Is it clear? And then from the sugars towards the inside is the bases. Is it making more sense to you now? Aisha and Iram? Very good. And next thing, the nitrogenous bases project towards the inside. The nitrogenous bases project towards the inside from the sugar. Okay. Written? The nitrogenous bases project towards the inside. And you can also say that it is perpendicular, right? Can you see it is perpendicular to the backbone? Perpendicular means 90 degrees. So bases are perpendicular to the backbone. Project inside. And you know that there is a rule. I have told you already about this rule, I think, right? Did I tell you about pyrimidine, purines? One purine binds with one pyrimidine only. Yes? Yes or no?
Yes.
Yes. Okay. Very good. So remember that one purine always binds with one pyrimidine, which is A binds with T and G binds with C all the time. Now, one more another important thing is, so when so I think you can imagine DNA as a right-handed coiled helix, right? So when it is coiled, just you can imagine this as a, have you ever seen a spherical staircase going up? Aisha,
Yes.
yes. So this is a spherical staircase. It takes one full turn to reach exactly at this position, right? Exactly at this position. Can you see this is one complete 360 turn? If these are the stairs, let's say it's going like this, then after taking one 360 turn, you will ascend from here to here, right? Probably one floor, let's say. Similarly, imagine a DNA. So can you see that from this point to this point is one turn, right? A 360-degree turn, one rotation of the DNA. If we imagine it like this, like a ladder uncoiled, then to coil it once, it is one 360-degree turn, yes? Yes or no?
Yes.
This is called the pitch of DNA. Okay. One 360-degree turn of the helix. In this case, it's the double helix. That's called the pitch. And in the pitch, there are around 10 base pairs. So base pairs is, this is one base. Let's say this is A, then this is T, this is G, and this is C. So here it will be T, A, C, G, right? So these two are pairs adjacent. So here you can see there are 10 base pairs in a pitch. And the length of the pitch is 34 nanometer. Sorry, 3.4 nanometer. Okay, this is the length of the pitch of DNA. So from here to here, it is 3.4 nanometer. So if I ask you now, what is the distance? Because this can only be calculated. You cannot visualize it. You cannot see it because it's a very, very small length. So if I ask you, what is the distance between two adjacent base pairs in a DNA? Can you tell me? I gave you all the information on the board. Think about it and tell me. Yes, Iram. Are you answering?
3.4.
3.4 is the distance of the pitch. The whole pitch is 3.4 nanometer. Let me make it very clear. 3.4 nanometer is the length of pitch from here to here. It is 3.4 nanometer. And I'm telling you, one pitch contains 10 base pairs. Okay. The lines that you see 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So there will be 10 base pairs in one pitch. And the pitch is 3.4 nanometers. So what will be the distance between two adjacent base pairs? Yes, no, something, tell me.
3.4 divided by 10.
3.4 divided
by
by 10. Yes, perfect. What is 3.4 divided by 10, Aisha? Great, good.
0.34.
Yes. So write down the distance between, this is calculated. This is how they figured out. Write down the distance between two adjacent base pairs in a DNA is 0.34 nanometer. Remember the unit, nanometer. If they ask in angstroms, then it will be 34 angstroms, because angstrom is nano. What is nanometer? Nanometer is 10 raised to power minus 9, and angstrom is 10 raised to power minus 10. So you will just remove 1 0. So 0.34 nanometer is 34 angstrom. Sorry. 3.4 angstrom. And 3.4 nanometer is 34 angstrom. Is it clear?
Yes.
Clear, yes or no?
Yes, sir.
Okay. And this, everything that you have studied, this whole thing, this double helix structure is called bDNA. In biology, the most common DNA is bDNA. So other forms of DNA, type of DNA is bDNA. So bDNA has a 10 base pair in between the pitch. For bDNA, the pitch is 3.4 nanometer or 34 angstrom. For bDNA, the distance between adjacent base pair is 0.3 nanometer or 3.4 angstroms. There are other types of DNA as well, which is not in your syllabus, aDNA, zDNA, etc. So those you don't have to study, okay? Don't worry about that. So I think that's all that we will do in this chapter. We'll finish here. Just note down everything. Have you noted everything down? Aisha and Iram? Okay, Iram? Cool. So in the next class, please read a little bit of morphology. We'll start with morphology. Which is your first chapter, morphology or anatomy? What do you want me to start with? Tell me.
G.
Both, Iram? Iram, you also want me to start with morphology?
Yes.
Okay, cool. So we'll start with morphology in the next class. And the last part of this chapter, enzyme kinetics, we'll do later on. Till here, do you have any questions, any queries, anything you want to ask?